Okay, don't touch the screen. Our screen touch chekanamma. Right. So autoimmunity. Here, immunity is a system that protects ourselves from various diseases or various pathogenic organisms. Sometimes, in some instances, some cases, our own immune system, it cannot identify our own antigens. You should remember one thing, that all of our cells, and our body fluids, they contain millions of proteins which are actually antigens according to the immune system or immunity. But sometimes, due to various reasons, okay, so our immune system, it loses its ability of tolerance. Okay, so what is it innate autoimmunity? The failure of tolerance or recognition ability of self and non-self discrimination. Okay, so that is the reason when our own immune system reacts on our own antigens or on our own tissues. So such phenomena is called as autoimmunity. So here there is no role of the innate immunity. So only T cells and the antibodies, that is the parts of the acquired immunity, participate in the autoimmunity. Okay, so here some are organic specific autoimmune. Just some diseases are given. Okay, so let's discuss some autoimmune diseases. Okay, so yeah, so in the one discussion, lymphocytes recognize some self antigens as foreign. Okay, so that results in production of autoantibodies and anti-response by T cells. Okay, so then, so number one, type 1, this is actually type 1. Okay, T1, diabetes mellitus. So it generally, it is seen, it is a kind of an autoimmune disease. Okay, it is seen in youth. Okay, so for various reasons. So due to last లో వస్తాయమ్మా the reasons for autoimmunity, కొన్ని చాలా తక్కువ రీజన్స్ ఉన్నాయి. సో ఇందులో ఏమవుతుంది, what happens in this type 1 diabetes mellitus? So insulin producing cells, okay, are attacked by self antibodies or self T cells. Okay, the pancreatic cells, okay, their receptors structures, their surface membrane proteins structure is altered. So the altered antigens or proteins are recognized as foreign, so by our immune system. And in type 1 diabetes mellitus autoimmune disease, self antibodies react on pancreatic insulin producing cells. Okay, so beta cells, they are attacked by our own antibodies. So that causes damage to damage to our pancreatic organ. So that causes type 1 diabetes mellitus. What happens? Insulin is not produced. Simple. Okay. So cytotoxic T cells first attack our altered pancreatic cells. Then macrophages also attracted to that area due to agglutination of antibodies. Okay, first autoantibodies bind to self antigens. Okay, so thinking that they are foreign, then cytotoxic T cells are attracted, then macrophages are attracted. Okay, so that leads to antibody dependent cell mediated cytotoxicity. Okay, so yeah, antibody mediated cell mediated cell lysis. This is cell lysis, antibody mediated cell lysis. So overall, this is a kind of మీరు ఈ దీనిలో ఒకటే పాయింట్ గుర్తుంచుకోవాలి ఏంటంటే ఇట్ ఈస్ అన్ autoimmune disease. Reason what? Okay, so due to the change of surface antigens of pancreatic insulin producing cells, okay, antibodies are produced against our own cells. So then due to that, the antibodies will form some antigen antibody complexes surrounding our pancreatic cells. So these cells are attracted. So antibody mediated cell mediated cytotoxicity or antibody mediated cell lysis takes place due to cytotoxic T cells and macrophages. Then another disease, Hashimoto thyroiditis. The name itself is indicating that. So here we all know that thyroid gland produces thyroxine hormone. Okay, to produce thyroxine hormone, it requires thyroid stimulating hormone. Okay, so thyroid stimulating hormone.
Sir, your voice is breaking, sir.
Okay, share up this. Right. Thyroid stimulating hormone, generally, how it works. For example, if it is a thyroid cell, generally some thyroid stimulating receptors are present like this. Okay, so to this thyroid stimulating hormone will come and bind to these receptors. Okay, in normal case. So that results in production of thyroxine hormone. Right. So here what happens, these receptor structure is modified. So antibodies are produced against అంటే ఈ రిసెప్టార్స్ కి ఆపోజిట్ గా యాంటీబాడీస్ అనేటువంటివి సెల్ఫ్ యాంటీబాడీస్ అనేటువంటివి వచ్చి ఇక్కడ అటాచ్ అవుతాయి. సో దట్ స్టిమ్యులేట్స్ continuously this thyroid cell to produce thyroxine. Immature thyroxine is produced. Okay, so due to continuous stimulation, after some time the cell may not work, so thyroxine lacks. So that is Hashimoto thyroiditis disease. It is a kind of autoimmune disease due to change in the structure of the thyroid receptor antigens or proteins. Okay, it is very common in women. Nowadays it is very, very common. Most of the women after the onset of at the age of 35 or 40, they are suffering from thyroid problem. So this is one such kind. Due to production of autoantibodies and sensitized T helper cells 1 against autothyroid antigens. Okay, so it causes hypothyroidism. After sometimes thyroxine production lekapena, even though there is no thyroxine ready to be released, okay, the thyroid cells are stimulated, but thyroxine is not secreted, that is not ready. So after some time or some period of time or after some days, the cells becomes non-functional, so thyroxine is not produced. That leads to hypothyroidism. This is another kind of another example for autoimmune disease. And this is very, very important, Amma. It is asked many exams. Chala, it was asked in many exams. Idi గుర్తుంచుకోవాలి మీరు. Okay, so what happens here? Myasthenia gravis. Recently, we attended an exam. So in that also, this question was asked. Even I was confused between the Hashimoto-thyroiditis and myasthenia gravis. రెండు ఆప్షన్స్ ఇచ్చారు. కొద్దిసేపు తల బుర్ర బద్దలు అంటే మర్చిపోవడం జరుగుతుంది. సో వాటిని చూడక చాలా రోజులైంది. సో బట్ లేటర్ వి పుట్ ఇట్ కరెక్ట్. ఎనీవే, సో కన్ఫ్యూజ్ కాకుండా నేను గుర్తు చేస్తున్నాను. Myasthenia gravis. What happens in myasthenia gravis? Auto antibodies are against acetylcholine receptors. So do you know acetylcholine receptors? I'll just try to draw some smaller diagram. So here in synaptic areas, okay, so the stimulus should reach from one presynaptic neuron to postsynaptic neuron. How it is reached? So by the chemical mediator called acetylcholine. This acetylcholine is released into this synaptic cleft. This is called synaptic cleft. This is presynaptic nerve. This is postsynaptic nerve now, okay, or this is one neuron, this is another neuron. So this is the place where they meet. Okay, so here in this area acetylcholine is released. So what does it do? So here on the postsynaptic neuron, some receptors are present. మళ్ళీ ఫిజియాలజీ వస్తుంది, బట్ మీకు ఐడియా ఉండడం కోసం చెప్తున్నాను, మర్చిపోయి ఉంటారు, అందరికి తెలిసి ఉంటుంది, రైట్. సో దీస్ acetylcholine receptors, they are the receptors for these released acetylcholine molecules. ఓకే, అవి వెళ్ళి ఇక్కడ బైండ్ కావడం, they will go and bind here, and this postsynaptic neuron will be excited and depolarization takes place. So that is how general process. But what happens here? Here, these the structure of these receptor molecules will be altered. So due to that, what happens? Our first thing, the acetylcholine cannot recognize these receptors. Okay, so the receptor structure is changed. So like this, so it cannot bind here, acetylcholine. So now the immunity comes into the picture. Okay, so it identifies this altered receptor, which is a protein, as a foreign antigen. So antibodies are produced against these receptors. So that stimulates or suppress the postsynaptic nerve. So that leads to some neurological problems. So in which the stimulus cannot be transmitted from one neuron to another neuron. Okay, so that is how the whole thing occurs in myasthenia gravis. Okay, production of autoantibodies against acetylcholine receptors present on motor end plates, particularly. So it occurs particularly. Just wait. What is motor end plate? So this is the nerve ending. Okay, so this is a skeletal muscle. So this area is called motor end plate, where that means neuromuscular junction is called motor end plate. So in that area, the acetylcholine receptors, they are modified, That leads to production of self-antibodies. Okay, so that leads to tissue damage. Okay, so what happens? The TH cells are accumulated there. They identify as foreign, and they stimulate B lymphocytes. B lymphocytes are modified into plasma cells, and antibodies are produced. They are attached to these modified receptors, and phagocytosis takes place. Cell cells are damaged or eaten. So tissue damage takes place. So these antibodies prevent acetylcholine to bind, and also complement mediated lysis takes place. Okay, so antibodies, they are attached to receptors. They prevent acetylcholine to bind there, and also the complement proteins will bind to these bound antibodies. So they act as opsonins. Okay, so that leads to opsonization, that is phagocytosis of cells. Okay, so due to this, some problems like weakening of muscles, okay, kallu mutra, drooping of eyes takes place, and inability to retract corners of mouth. Okay, so and nose తెరిస్తే మళ్ళీ మూసుకోవడం ఇబ్బంది అవుతుంది, are some early symptoms. Okay, corticosteroids and immunosuppressants are used to control. Okay, so anyway, మీరు ఇక్కడ ఏం గుర్తుంచుకోవాలి? ఒకటి, the antibodies are produced against acetylcholine receptors, that is myasthenia gravis, as simple as that. Okay, then systemic lupus erythematosus. Okay, it is simply called as SLE. Okay, so ఇది కూడా 2-3 సార్లు అడిగాడు. It is example for systemic autoimmunity, more common in women. So the 10 మందిలో 9 మంది ladies కి వచ్చే అవకాశం ఉంది. So auto antibodies are produced against our DNA, histone proteins, RBCs, platelets, WBCs, and clotting factors. ఇక్కడ mostly related to the DNA of the blood cells and RBCs, platelets, all these, all their antigens are modified. So due to that, same process takes place. So our immune system recognize them as foreign. Okay, so antibodies are produced, our cell mediated immune cells are reached there. They will kill, they will eat. Okay, they will damage our own blood cells, and the blood vessels or capillaries. Okay, so due to that, skin rashes, fever, weakness, kidney dysfunction, and arthritis are major symptoms of this systemic lupus erythematosus. Hemolytic anemia, thrombocytopenia, vasculitis. Vasculitis means damage of blood vessels may be observed. It also occurs in type 3 hypersensitivity. Same thing is seen in next topic, type 3 hypersensitivity. Okay, so that is about the systemic lupus erythematosus. So దీన్ని బట్టి it is related to blood అని ఒకటి గుర్తుంచుకోండి. Okay, so then ఇక్కడ ఏమవుతుంది, mostly blood related problems are occurred. So internal bleeding takes place. Okay, దానివల్ల skin rashes వస్తాయి, that is main. Okay. Right, so then multiple sclerosis. Actually, this is name is a little bit confusing. It is due to autoreactive T cells against myelin sheath of neurons. So multiple sclerosis అనే డిసీజ్ ఏమవుతుంది, autoreactive T cells are produced against our own neurons, particularly the myelin sheath also contains some antigens. Okay, so their structure is modified. Okay, so our immune system recognize as foreign, and action is taken by the immune system, particularly myelin sheath of neurons of brain and spinal cord. Okay, so their myelin sheath antigens are modified. So that causes many production of antibodies and immune cells are released and myelin sheath is damaged. Okay, so that causes many neurological disabilities like paralysis, loss of vision, etc. So that is different kinds of neurological problems are taken. That's why it is multiple sclerosis. Okay, so it is also frequently occurs in 2 to 3 times greater than the men. Right. So then rheumatoid arthritis, it is mostly, it is also seen in women mostly. So a kind of arthritis related to joint pain. Okay. Another most common autoimmune disease. It is seen in the women between the age 40 to 60 years aged, predominantly women. Autoantibodies are called here rheumatoid factors. Why? So example, IgM is produced against our own IgG to form immune complexes deposited in joints. Okay. ఏమవుతుంది అంటే here IgG నేను మీకు చెప్పాను ఏంటంటే ఈవెన్ యాంటీబాడీస్ ఆర్ ప్రోటీన్స్ దే ఆల్సో కంటైన్ సమ్ ఎపిటోప్స్. ఓకే, సో వెన్ ఇన్ autoimmune conditions, okay, the self epitopes are modified. Okay, so the structure is changed, so they are recognized as foreign molecules, and IgM is produced against uh, IgG antibodies to form immune complexes. These immune complexes, they are deposited in joints. Gout disease lagane oka rakanga. So leading to complement activation causing type 3 hypersensitivity. Okay, so immune complex mediated hypersensitivity ni type 3 hypersensitivity antam. Ikkada kuda immune complexes అనేటువంటి ఫామ్ అయినాయి, ఎక్కడ జాయింట్స్ లో ఫామ్ అయినాయి. ఓకే, సో దానివల్ల ఏమవుతుంది అని అంటే అక్కడ ఆప్సనేషన్ జరుగుతుంది. ఓకే, అండ్ దట్ మీన్స్ ఫాగోసైట్స్ వచ్చి ఆ ఇమ్యూన్ కాంప్లెక్సెస్ ను డిస్ట్రాయ్ చేసే క్రమంలో, సో ఇన్ దట్ ప్రాసెస్ అవర్ జాయింట్స్ ఆర్ ఆల్సో డ్యామేజ్డ్. ఓకే, అండ్ క్రానిక్ ఇన్ఫ్లమేషన్ అక్కడ. సో వెనెవర్ ద న్యూట్రోఫిల్స్ అండ్ ఫాగోసైట్స్ ఇన్వాల్వ్, ద ఇన్ఫ్లమేషన్ is very very common. So that leads to severe joint pain. Okay, so movement becomes very painful. Okay, so for these non-steroidal anti-inflammatory drugs are used. So what you have to remember is, so it is IgM is produced against IgG and immune complexes are formed. They are stored in joints. That leads to opsonization, okay, and tissue damage, particularly the joints area. Okay, so our immune cells will go and damage our own joint area tissues. Okay, so that is another example for autoimmune disease. So causes, what are the various causes for the autoimmune diseases? There are many factors. What is here, these two are same. Geographical area. So here, what does it mean? It means that pollution. Okay, so in industrial areas, due to inhalation of many carcinogenic harmful chemicals. Okay, so in the food, water and air, so that those chemicals, they alter our DNA structure. That leads to alteration of deformed antigens, self antigens, or deformed proteins or abnormal proteins are formed. So that leads to autoimmunity. That is one of the reasons. And infections. So some viruses also can cause mutations in our DNA that leads to altered protein production. Okay, so dietary habits, lifestyles, some food additives. Some food additives can also damage our they are just like drugs. Okay, so in recent times, in this fast food culture, some food additives which are present in our foods, Okay, they also change, alter the self antigen structure, or they may cause mutations in our DNA. Okay, lifestyle like smoking. Okay, are some of the extrinsic factors. These are all extrinsic factors from geographical area to lifestyle. So here, intrinsic factors like gene expression, which involves in growth and development of cells and organs. Okay, so here gene expression abnormal gene expression also one of the factors for autoimmune disease. People with HLA-B27 allelic expression are 90 times more chance of getting ankylosing spondylitis, a kind of neck pain. Okay, so related to backbone, right? So this is a gene, HLA-B27 gene. So there are different kinds of MHC genes are present, which we discussed. Okay, so among them, if HLA-B23 is present, okay, there is more chance of occurrence of this ankylosing spondylitis. Okay, so that change the cell surface antigens. Okay, and another gene, AIR gene, is important to code for a protein that removes autoreactive T cells in thymus. Okay, if mutation occur, okay, if in the AIR gene, if at all any mutation occur, okay, this disease is so autoimmune poly ఇది ఫస్ట్ టైం నేను కూడా పాలి enocrinopathy. Okay, candidiasis, ectodermal dystrophy. Okay, the first time. Okay, the APECED is a systemic disease that occurs. Okay, so when ARI, that means related to some thymus gland disease. Okay, so and IPEX, immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. It is also occurred due to mutation in AIR gene. Okay, so it is sorry, not AIR. It is caused due to mutations in FOXP3 gene, which is important for expression of T regulatory cells. Okay, one is this AIR gene, which codes for proteins of code for a protein that removes autoreactive T cells. అంటే మన సొంత యాంటీజెన్స్ ని డ్యామేజ్ చేసేటువంటి టీ సెల్స్ ని రిమూవ్ చేసే ప్రోటీన్. ఓకే, ప్రోటీన్ దట్ రిమూవ్స్ autoreactive T cells in thymus. Okay, so that protein is coded by AIR gene. Okay, AIRE gene. Okay, if at all mutation occurs in AIRE gene, okay, so that leads to APECED, autoimmune polyencephalopathy. Polyencephalopathy, candidiasis, ectodermal dystrophy is a systemic disease occurs due to mutation in AIRE gene and a mutation in FOXP3 gene, which is important for expression of T regulator cell. What is T regulator cell? TH and T suppressor cells. Okay, so what expressions ki e FOXP3 gene యొక్క protein అవసరం. If at all the mutation is occurred in this FOXP3 gene, okay, so this immune dysregulation, endocrinopathy, enteropathy, and X-linked syndrome, it is occurred. So a kind of immune related disease. Okay, just so these are some of the autoimmune diseases. Okay, there are examples for the autoimmunity. So autoimmunity is a failure of tolerance or recognition, a failure of recognition ability of self and non-self uh, discrimination power by our immune system. Okay, famous examples are Hashimoto thyroiditis and myasthenia gravis, systemic lupus erythematosus, and then rheumatoid arthritis. These are the very, very famous auto uh, immune diseases. Okay. So that is about the autoimmune. So our next topic will be hypersensitivity. Are you able to see this screen? కనబడుతుందా?
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
Okay, fine. Hypersensitivity కనబడుతుందా?
Yes, sir.
Okay. Sure. ఫేర్ చేయలేదు, నేను మీకు నోట్స్ పంపేటప్పుడు ఫేర్ చేసి పంపుతాను. జస్ట్ నేను నన్ను ఫాలో అయితే హైపర్ సెన్సిటివిటీ ఇది నేను ఎలా రాసుకున్నానో అలాగే, అంటే జనరల్గా నేను నోట్స్ రాసిన తర్వాత పీపీటీలో ఎడిట్ చేస్తాను, బట్ నాకు టైం లేక దీన్ని ఎడిట్ చేయలేదు. ఎనీవే రైట్. సో వాట్ ఈస్ హైపర్ సెన్సిటివ్? హైపర్ సెన్సిటివిటీ ఈజ్ అదర్వైజ్ ఆల్సో నోన్ యాజ్ అలర్జీ. Okay, so it is not, and it is related to autoimmunity, but not autoimmune reaction. Okay, so what is the difference between autoimmunity and hypersensitivity is our self tolerance is lost in the case of autoimmunity. Okay, so our immune system, it cannot identify our own cells or self antigens. But whereas in hypersensitivity, our immune system functions more than normal. Okay, overexpression of immune system is hypersensitivity. Okay, so you know, its another name is allergy. So allergies means, allergy means altered, eron means action. Altered action is allergy. Okay, so this allergy term was first used by one request. Okay, so here what happens in hypersensitivity, either due to repeated exposure of a single antigen. Okay, so mostly are already sensitized, cells, if at all already sensitized cells are present. Sometimes only one time, if at all they are sensitized. In the second case, they react very aggressively. That leads to tissue damage. Okay, hypersensitivity lo mostly tissue damage jarugutundi. Okay, so such second dose ni manam shocking dose or eliciting dose అని అంటాం. Anyway, ఇక్కడ hypersensitivity, it includes 3 steps. One is sensitization, that means first entry of antigen, waiting period. Okay, so in this period, after sensitization, antibodies are produced, or our immune cells will become ready. Whenever the same antigen is given or entered next time. Okay, so the reaction of the immune system is very fatal, very aggressive, very harmful, sometimes in some cases. Okay, so that leads to hypersensitivity. Okay, these are the 3 steps which are involved in hypersensitivity. So these 2 scientists, Coombs and Gell, are the scientists who properly classified the hypersensitivity. Earlierly, the hypersensitivity was divided into 2 types. One was immediate type of hypersensitivity, another one is delayed type of hypersensitivity. So first three were added in immediate, and last one is generally called as both in this classification and normal classification as delayed type of hypersensitivity. Anyway, according to Coombs and Gel, there are four kind of hypersensitivity. Okay, they are type 1, type 2, type 3, type 4 hypersensitivities. Simple as that. Okay, there are four kinds of hypersensitivity reactions. Okay, they are simply type 1, type 2, type 3, and type 4. Okay, so type 1, there are different names for type 1 hypersensitivity reaction. Okay, so that is the type 1 is generally called as anaphylactic hypersensitivity. It is also simply called as anaphylaxis. Okay, so it is also known as the most rapid immediate type of hypersensitivity. అన్నిటికన్నా fast గా జరిగే hypersensitivity type 1. You remember. Okay, so ana means anti-phylaxis. Okay, means protection. So here the anti-reaction takes place or against the action is taken place against the protection, that is anaphylaxis. Okay, so it was identified by Richard in 1902, right, or described by Richard in 1902. So he conducted some experiments in dogs. So with a glycerin which was extracted from sea anemones, okay, he injected such glycerin to dogs ఇవ్వడం జరిగింది. So in first case there was no reaction against that glycerin, and in second dose, eliciting dose or shocking dose, okay, within minutes the dog was died. Okay, same experiment also done in guinea pigs. In guinea pigs with egg white protein. Okay, so dogs lo nemo sea anemone glycerin to cheyadam jarugindi. In guinea pigs he injected egg white protein. Okay, so the result is same. So what happens, this is beautiful explanation. Type 1 or immediate type of hypersensitivity, whenever some special type of antigens they enter into our body, any antigen is entered into our body. Okay, so antigen presenting cells, they engulf the antigen. So here these are the antigens. Okay, this is antigen presenting cell, APC. Okay, so this is T helper cell. Okay, so whenever the antigen entered into this antigen presenting cell, it processes, it exhibits, or it presents some part of the antigen, okay, to the T helper cell. Okay, they're excited or activated. T helper cell 2s are produced. They release some cytokines, okay, that stimulate B lymphocytes. These B lymphocytes are divided and differentiated to form plasma cells. Plasma cells produce antibodies. Here, these antibodies are special type. Generally, normally IgM type of antibodies are produced, but to some antigens, maybe due to their different structure, whatever it may be, here for the first time itself, IgE kind of antibodies are produced. Here, I told you during the cells of immune system. Okay, mast cells and basophils contains some receptor proteins for IgE antibodies. Okay, mast cells and basophils, they contain on their membrane some receptor proteins, okay, for the IgE kind of antibodies. So these, for the first time, when the antibodies are released first time, okay, these antibodies will go and bind to these special membrane bound antibody receptors. So ఇవి వచ్చి ఇక్కడ అతుక్కోదు. So this is first case, there will be no reaction. Okay, so in re-exposure phase after 10 days, okay, or 5 to 10 days, he injected the same kind of the antigens to the animal. Okay, so same reaction taken place, but here these reactions are not occurred. Okay, these are generally identified by activated B lymphocytes. Okay, that is, they are memory cells. Memory cells will observe these antigen. Okay, they engulf. Okay, so they are automatically activated. Okay, with the help of T helpers into, they are activated, and they will be differentiated into plasma cell. Okay, plasma cell second time produce what we call the antibodies. These antibodies again they are attached here, and some of the antigens. Okay, here the distance has reduced here. So the reduced distance are the antibodies are joined by these antigens. Okay, so that shockingly or very aggressively excite the mast cells or basophils, which release some granules in their vicinity. Okay, so these granules includes histamines, bradykinins, serotonin, like allergic substances, okay, which are released more in number. Okay, that leads to uh, various actions. See, toxides, vaccines, trypsin, insulin-like proteins, dextrans, barbiturates, folic acid, streptomycin, thiamine. Okay, foods like crabs, fishes, egg white, milk, lobster, shrimps, snake, and wasp venoms. Okay, so all these are the antigens, a different kind of antigens. They cause the immediate type of hypersensitivity. In the re-exposure or booster dose or shocking dose, okay, so these antigens will go and bind or join these antibodies that excites these mast cells to release, to release histamine, serotonin, okay, eosinophil chemotactic factor, okay, A, and neutrophil chemotactic factor A, heparin, etc. These are called primary mediators. Okay, and due to their action, prostaglandins, leukotrienes, thrombokinase. Okay, so TAF, this is, I forgot its name, EAF, and bradykinin, cytokinins. All these are the secondary mediators. They are released. What they do? So they do vasodilation, one of the action. So that means expansion of blood capillaries takes place. So that leads to sudden dip in BP. బీపీ అనేది సడన్ గా పడిపోతుంది, ఎందుకంటే బ్లడ్ వెజల్స్ అనేటువంటి ఎక్స్పాండ్ కావడం వల్ల. సో బ్లడ్ ప్రెషర్ అనేది తగ్గిపోతుంది. సో డ్యూ టు దట్ సడన్ low BP, it causes shock. Okay, so that leads to various clinical manifestations. Okay, sometimes the animal actually died in these two cases. Okay, so these factors, so they cause reddening of skin, and some rashes are occurred. జనరల్ గా మీరు బయట కొంతమందికి అలర్జీ అవుతూ ఉంటుంది కొన్ని పడకపోతే. సో బాడీ పైన ర్యాషెస్ కావడం అనేది టైప్ 1 హైపర్ సెన్సిటివిటీ. వాళ్ళు తినకూడదు ఏదైనా తిన్నా, ఓకే, సో డస్ట్ పడకపోయినా ఇమీడియట్ గా ర్యాష్ వస్తుంది. సో దట్స్ వై దట్ ఈస్ ఇమీడియట్ టైప్ ఆఫ్ హైపర్ సెన్సిటివిటీ అంటాం. ఓకే, సో దిస్ ఈస్ మెయిన్ థింగ్. ఇరిటేషన్ ఇన్ ట్రాకియా అండ్ బ్రాంకై, allergy in our respiratory passage is observed that leads to rhinorrhea, that means continuous fluid leakage from nose. Okay, so and that leads to asphyxiation. Okay, so that means unable to breathe anamata. Okay, and abdominal cramps, sometimes due to this the person may die due to asphyxiation and the abnormal cramps. Okay, and the vomitings and the motions are also occurred, convulsions, okay, abnormal behavior. So these are all the symptoms of anaphylaxis, that is type 1 hypersensitivity. First example is anaphylaxis. Second example is due to anaphylaxis, another kind of immediate type of hypersensitivity is atopy or atopic allergy. It was explained by a scientist called Koka in 1943. So it occurs due to natural antigens. Okay, so it is seen ఇందాక నేను చెప్పినటువంటిది దీనికి వస్తుంది, డస్ట్ ఎలర్జీ కానీ ఏదన్నా యూనో ఎగ్ వైట్ తినడం కానీ సబ్ ఫుడ్స్ తిన్నప్పుడు కానీ జరిగేది అటోపీ అన్నమాట. సో న్యాచురల్ దాదాపు 10% ఆఫ్ ది పాపులేషన్ ఆర్ సఫర్డ్ విత్ ద అటోపీ. ఓకే, సో ఇన్హలేషన్, ఇంజెక్షన్ ఆర్ కాంటాక్ట్ సబ్స్టెన్సెస్ లైక్ హే అంటే దీన్ని మనం తెలుగులో చొప్పు అంటాం. ఓకే, సో డ్రైడ్ బ్రాంచెస్ ఆఫ్ paddy and corn, that is called hay. Okay, pollen grains, okay, dust, feathers of birds, hair, spores of fungi, okay, cockroach, dung, and insect excretory substances also. Whenever they are inhaled or ingested into our body, or sometimes due to direct contact, okay, they enter into our skin, okay, through our skin. Okay, foods like seafoods, crustaceans, okay, and egg white is also one of the example. Okay, that leads to allergy. అవెళ్ళి సింపుల్ గా మాస్ట్ సెల్స్ పైన ఉన్నటువంటి ఆ యొక్క మెంబ్రేన్ బౌండ్ యాంటీబాడీస్ ఆర్ జాయింట్ బై దీస్ సబ్స్టెన్సెస్. ఓకే, వీటిలో ఉన్నటువంటి స్మాల్ పార్టికల్స్ వెళ్ళి ఆ యాంటీబాడీస్ ని జాయిన్ చేస్తాయి అన్నమాట. ఇలా ఇవి ఆ సబ్స్టెన్సెస్. Okay, so that causes allergy, atopic skin allergy అవుతుంది. Okay, so it causes hay fever, a kind of fever, asthma, so problems in breathing, and atopic dermatitis, ఇందాక చెప్పినటువంటి ర్యాషెస్ అనేటువంటి అవుతాయి. సో దట్ ఈస్ మీరు టైప్ 1 హైపర్ సెన్సిటివిటీలో పార్టిసిపేట్ చేసే యాంటీబాడీ ఏంటి? ఐజీ ఒకటి. విచ్ సెల్స్ పార్టిసిపేట్ release the hypersensitivity granules, mast cells and basophils. Which is the most rapid hypersensitivity, type 1. Okay, so and yeah, ఈ రెండు హెడ్డింగ్స్. ఒకటి అనఫైలాక్సిస్ అనేది నెంబర్ 1. నెంబర్ 2 ఏంటి అంటే అటోపిక్ అలర్జీ ఆర్ అటోపీ. ఈ రెండు హెడ్డింగ్స్ గుర్తుంచుకోవాలి. రైట్, సో దట్ ఈస్ అబౌట్ టైప్ 1. And type 2 hypersensitivity. It is otherwise also called as antibody dependent cell mediated cytotoxicity. దీన్ని షార్ట్ ఫామ్ లో ఏమని పిలుస్తాము అంటే ఏడిసిసి. సో క్యాప్స్ లో రాయాలి. సో హియర్ it is type 2 hypersensitivity is also known as ADCC, or antibody dependent or antibody mediated cell mediated cytotoxicity. Cyto cell toxicity. So harm. Okay, due to some toxic substance. Okay, your cells అనేటువంటివి డ్యామేజ్ అవుతాయి. Okay, so due to antibody. Okay, our own antibody. Right. Here, membrane bound antigens like blood group antigens like A, B, RH, thyroid antigen, okay, and cedar-made antigen. Cedar-made is a drug, okay. So for this drug, some receptor molecules are present on platelets. So ఇవన్నీ కూడా మనం membrane bound antigens అంటాం. ఏంటి membrane bound antigens? The blood group antigens like, antigen A, B, RH antigen, thyroid antigen, okay, which is present on thyroid cells, and cedar-made antigens of platelets. All these are called membrane-bound antigens, self-antigens. Okay, so here complement system play an important role in this kind of antibody-mediated, what we call the cell-mediated cytotoxicity. Okay, so C3b, a complement protein that acts as opsonium. ఇక్కడ ఏంటి అంటే ఈ బ్లడ్ గ్రూప్ యాంటీజెన్స్ కావచ్చు, RH యాంటీజెన్ కావచ్చు, థైరాయిడ్ యాంటీజెన్ మేబీ. ఓకే, సో హియర్ ద antibodies are produced, which are generally produced to these, against these, antigens which are present on different cells and different tissues. Okay, so here what are the examples? Okay, so one of the example, first example ఏంటి అంటే mismatched blood transfusions. నేను ఎక్స్ప్లెయిన్ చేయట్లేదు నేను. సో దే ఆర్ ఆల్సో నోన్ యాజ్ ఐసోఇమ్యూన్ రియాక్షన్స్ అనేది గుర్తుంచుకోవాలి. ఫస్ట్ ఎగ్జాంపుల్ ఫర్ టైప్ 2 హైపర్సెన్సిటివిటీలో ఫస్ట్ ఎగ్జాంపుల్ ఏంటి అంటే మిస్మ్యాచ్డ్ బ్లడ్ ట్రాన్స్ఫ్యూషన్స్. ఇది మళ్ళీ వస్తుంది జెనెటిక్స్ లో, అక్కడ మళ్ళీ డీటెయిల్ గా చెప్తాను. బట్ ఏమవుతుంది, సో జనరలీ హ్యూమన్ బీయింగ్ ఎగ్జిబిట్ IgA, antigen A, antigen B and RH antigen on RBC cell surface. Okay, whenever a person receives blood from a donor, okay, if at all the blood groups of both receiver or donor are similar, there will be no action. But if at all the antigens which are received or blood group blood which is received from a donor is not similar to the receiver's antigen type, then isoimmune reactions takes place. Here antigen A, antigen B are isoimmune antigens. Okay, so a person antigen A కలిగి ఉంటాడు, ఇంకో person antigen B కలిగి ఉంటాడు. So recipient immune system or blood contains if he contains antigen A in his serum, in his blood, antibodies are present against B antigen. So ఒకవేళ A blood group ఉన్న వ్యక్తికి, if at all B blood group is transfused, okay, so the antibodies which are already present in the recipient's blood or serum, okay, so they directly act on the received blood cells. Okay, that causes a kind of hypersensitivity. Antigen antibody complexes are clumped. Okay, so phagocytosis takes place. Okay, that leads to these antigen antibody complexes. They block the smaller capillaries. That leads to complete tissue damage. Okay, tissues కి blood reach కాదు. అప్పుడు ఏమవుతుంది, ఆ టిష్యూ అంతా బ్లాక్ కలర్ లో మారిపోతుంది. Cells gradually will, due to lack of oxygen and foodstuffs, the cells will be damaged or becomes become non-functional. And second example for type 2, ఇది కూడా నేను ఎక్స్ప్లెయిన్ చేయను. Hemolytic disease of
newborn.
Very good. ఇది కూడా I am not going to explain. Right. So it is also a kind example for an isoimmune reaction. So here a mother contains blood group Rh negative and a fetus contains blood group Rh positive. This HDN takes place. దీన్ని మనం ఇంకొక పేరు చెప్తారా ఎవరన్నా? What is another name for this?
Erythroblastosis.
Very good. The results కాబట్టి నేను దీన్ని ఎక్స్ప్లెయిన్ చేయదలుచుకోలేదు. Okay, this is second example. ఏమవుతుంది? So first time Rh antibodies are produced during the in the mother's immune system. So general ga rare ga RBCs are entered from fetus to mother. In such conditions, for the first time IgM kind of antibodies are produced in the primary immune reaction. So these IgM antibodies, they cannot cross the placental barrier. So in the first pregnancy time, due to as they cannot enter into fetal circulation by crossing placenta, so first child will be normal. Okay, when in the second pregnancy, okay, so as usual, the fetus blood group is Rh positive. So at that time, if at all some RBCs are entered into mother's circulation, okay, so due to leakage or something, so secondary immune reaction takes place. Memory cells quickly identify the Rh positive antigens and IgG antibodies are produced, which can cross the the placental barrier. So the IgG antibodies are released against the Rh positive antigens of fetus. So those will enter into fetal circulation and they will agglutinate the RBCs of fetus. That leads to severe hemolysis in the fetus. Okay, severe jaundice in the fetus. Sometimes death may also take place. So for this, antibodies are given. Okay, so for the mother, to prevent this hemolytic disease of newborn or erythroblastosis fetalis. ఇక్కడ ఏమవుతుంది అంటే యాక్చువల్ ఎరిథ్రోబ్లాస్టోసిస్ అని ఎందుకన్నారు అంటే in fetus all the matured RBCs are damaged due to the release of IgG from mother. అప్పుడు ఏమవుతుంది? The bone marrow and liver, particularly in the liver, tries to produce adequate amount of or number of RBCs. Okay, దానివల్ల ఏమవుతుంది? త్వరగా ప్రొడ్యూస్ చేయాలన్నా తొందరలో, okay, so due to rapid need, okay, immature RBCs are produced in the fetus. Those immature RBCs are erythroblasts. ఇది మళ్ళీ వస్తుంది హీమోపయాసిస్ చెప్పినప్పుడు స్టెమ్ సెల్స్ ప్రైమ్ లో వస్తుంది అండ్ జెనెటిక్స్ లో వస్తుంది. సో ఆ ఎరిథ్రోబ్లాస్ట్ అనేటువంటి ప్రొడ్యూస్ కావడం ఒకటి, హీమోలైసిస్ జరగడం, రెండు రిజల్ట్స్ వల్ల ఫీటస్ సఫర్ ఎలాట్. దట్ ఈస్ హీమోలైటిక్ డిసీజ్ ఆఫ్ న్యూబార్న్ type 2 hypersensitivity. And then here myasthenia gravis. Okay, it is also a kind of type 2 hypersensitivity. So here ఇందాక మనం అన్ని చూసినాం. ఇక్కడ ఏం జరుగుతుందంటే మనం ఏంటి డెఫినేషన్ ఏంటి అంటే మెంబ్రెయిన్ బౌండ్ యాంటీజెన్స్ కి అగెనిస్ట్ గా యాంటీబాడీస్ ప్రొడ్యూస్ అవుతాయి. ఓకే, హియర్ membrane bound antigen ఏంటి అంటే ACH receptor. Its structure is modified. Okay, so antibodies are produced against acetylcholine receptors, which is a protein, which is an antigen of neuron. Okay, so that leads to myasthenia gravis ఇందాక చూసినాం. And good posture syndrome ఇది కూడా చూసినట్టున్నాం. So here what happens, antibodies are produced against glomerular basement membrane. Okay, particularly epithelial cells. Okay, glomerulus అనేది కిడ్నీలో నెఫ్రాన్ లో ఉండేటువంటి పార్ట్, దానిలో అన్ని నెట్వర్క్ ఆఫ్ బ్లడ్ క్యాపిలరీస్ ఆర్ ప్రెసెంట్ విచ్ కంటైన్స్ basal membrane which is epithelial in nature. So they also contain antigens, they are modified, and alveolar epithelial cells. Okay, alveoli is the terminal part of the lungs which is made up of squamous epithelial cell. Okay, so their antigens are modified, so antibodies are produced against these basement membrane cells of glomeruli of kidney, nephron, and alveolar epithelial cells. వీటికి against antibodies produce చేయడం వల్ల, so this good posture syndrome is a kind of disease that causes problems in breathing or respiration and excretory function. And then Hashimoto thyroiditis, ఇందాకనే చూసినాం దీని గురించి. So the name itself is indicating that it is related to thyroid gland. So TSH receptor structure is changed, which are present on the cell membrane of the thyroid cells. So that antibodies are produced against those altered receptor proteins. So that leads to hypothyroidism, that is called Hashimoto thyroiditis. All these are autoimmune disease also, and also example for type 2 hypersensitivity. And last one is drug induced hypersensitivity type 2 లో. ఇప్పటి వరకు చెప్పుకున్నాయి autoimmune disease, HDN, and isoimmune reactions. Isoimmune reactions అంటే కన్ఫ్యూజ్ కాకండి. ఇది నెంబర్ 1. You should not confused. Okay, isoimmune reactions అంటే ఏంటంటే, so blood group reactions. Okay, isoimmune reactions. Second one, erythroblastosis fetalis, another example. Myasthenia gravis, good posture syndrome, Hashimoto thyroiditis అనేటువంటివి ఇవి థర్డ్ కేటగిరీ. ఓకే, సో ఫోర్త్ కేటగిరీ ఏంటి అంటే డ్రగ్ ఇండ్యూస్డ్ type 2 hypersensitivity. So some drugs, due to continuous usage of some drugs, they modify some antigens which are present on our own cells. So drugs like penicillin, phenacetin, or chlorpromazine, okay, which causes hemolytic anemia, pyridine, agranulocytosis, okay, sedamoid of thrombocytes or platelets, okay, so that causes thrombocytopenic purpura. So here all these diseases, agranulocytosis, that means so agranular cells are produced more in less in number. Okay, hemolytic anemia, that means our RBC antigens are modified. So and due to continuous utilization of these drugs like penicillin, phenacetin, or chlorpromazine, okay, that causes hemolytic anemia. Pyridine usage causes agranulocytosis. Okay, sedamoid is a kind of drug, okay, that causes thrombocytopenic purpura, that means lesser production of all these cells, platelets, agranulocytes, and RBCs, okay, due to drugs, usage of drugs more than normal. Okay, change the shape of the self antigen on RBCs, neutrophils, some platelets. Okay, so that causes the hypersensitivity, or antibodies are produced against those corresponding cells or our immune cells will go and attack our own cells. So to identify this, whom's test is conducted, adokati gurtunchukondi. It's not big thing. Type 3. So ఇది ఒక హెడ్డింగ్ గుర్తుంచుకోవాలని మీరు ఫస్ట్ హెడ్డింగ్ తర్వాత ఉన్న హెడ్డింగ్ గుర్తుంచుకోవాలి. సో type 2 is a antibody dependent cell mediated cytotoxicity. Okay, first one is anaphylaxis and atopic allergy and immediate type of hypersensitivity. Type 3 మనకి ఎగ్జాంపుల్ ఇదే అడుగుతారు, మిగతా అంతా పక్కన పెడితే ఇదే ఈ హెడ్డింగ్ అడుగుతారు. Immune complex mediated type of hypersensitivity is which type of hypersensitivity? Type 1, type 2, type 3. Type 3. Okay, so type 3 is immune complexes mediated hypersensitivity. Adokati baga gurtunchukovali, right? So then here what happens, some antigen antibody complexes are accumulated in tissues. Okay, then explain this. See here, in general, when an antigen is entered, antibodies are produced. Okay, antibodies will go and bind to antigen, surround them. Okay, so antigen antibody complexes are formed in large number. Those formed antigen antibody complexes are eliminated. Okay, so by engulfing or eating by the neutrophils and macrophages. Okay, that is general case. In some cases, the accumulated antigen antibody complexes, okay, they cannot be identified or they cannot be removed from the body or tissue. So that causes this type 3 kind of hypersensitivity. ఏమవుతుంది అంటే ఇందులో మెయిన్ గా ఐదర్ యాంటీజెన్ ఎక్సెస్ ఆర్ యాంటీబాడీ ఎక్సెస్. ఓకే, సో ఒక్కొక్కసారి ఏమైందంటే antigens number, if at all antigens number is more than normal titer value, okay, so the antigen antibody complexes cannot be cleared properly by macrophages. That is one case. Same case occurred whenever antibodies are produced more in number. Okay, so that is also case when macrophages fail to clear all the complexes. And okay, సో ఏమవుతుంది, ఆ కాంప్లెక్సెస్ కి C59 అనేటువంటి ఒక కాంప్లిమెంట్ ప్రోటీన్ అనేది వెళ్ళి యాంటీబాడీ హెఫ్సి పార్ట్ కి అటాచ్ అవుతుంది. ఓకే, సో ఐజీజీ వెళ్ళి ఈ కాంప్లెక్సెస్ కి అటాచ్ అయితే దాని యొక్క ఎఫ్సి పార్ట్ కి C59 అనేది వెళ్ళి అతుక్కుంటుంది, విచ్ యాక్ట్స్ యాజ్ అన్ ఆప్సోనిమ్, అంటే ఒక జెండా లాగా, ఒక సిగ్నల్ లాగా దేనికి, మాక్రోఫేజెస్. ఓకే, సో ఎప్పుడు అది, వెన్ నెంబర్ ఆఫ్ antigen antibody complexes are accumulated in some tissues, అప్పుడు ఈ C59 వెళ్ళి ఆ యాంటీబాడీస్ అతుక్కున్న తర్వాత మాక్రోఫేజ్ వచ్చి ఆ కాంప్లెక్సెస్ తో పాటు అక్కడ అండ్ దే కెనాట్ డిస్క్రిమినేట్ at that moment. Okay, excess of antigens ఉండడం వల్ల, so they even eat our own cells, that leads to tissue damage. Okay, so immune complexes అనేటువంటి క్లియర్ చేయడంలో ఫెయిల్ అవుతుంది ఇమ్యూన్ సిస్టమ్. ఓకే, సో టిష్యూ డ్యామేజ్ డ్యూ టు ఫాగోసైటోసిస్. ఓకే, ఇక్కడ ఏమవుతుంది, C59 ఆప్సోనిన్ పనిచేస్తుంది, ఆప్సోనిన్ కి అట్రాక్ట్ అయ్యి ఫాగోసైట్స్ వస్తాయి, ఫాగోసైట్స్ వచ్చి ఆ ఇమ్యూన్ కాంప్లెక్సెస్ రిమూవ్ చేసే క్రమంలో దే ఆల్సో ఎంగల్ఫ్ ఆర్ ఈట్ అవర్ ఓన్ టిష్యూ సెల్స్. Okay, so there are two kinds of famous experiments, or what we call the reactions in type 3 hypersensitivity reaction. Okay, so one is Arthur's reaction, serum sickness. Ide adutaru in example. Arthur's reaction is an example of type 3 hypersensitivity. Serum sickness is an example of type 3 hypersensitivity. Okay, so మీకు అర్థం కావడానికి కొన్ని పాయింట్స్ యాడ్ చేశాను కానీ సో ఈ హెడ్డింగ్స్ గుర్తుంచుకోవాలి కంపల్సరీ. Okay, so Arthur's reaction is also known as antibody excess kind of hypersensitivity. Okay, antibody excess అంటే దీని అర్థం ఏంటి? ఆర్థర్స్ రియాక్షన్ లో యాంటీజెన్స్ కన్నా యాంటీబాడీస్ నెంబర్ అనేది చాలా ఎక్కువ మోతాదులో ఉంటుంది. ఓకే, ఇట్ వాజ్ ఐడెంటిఫైడ్ బై నికోలస్ ఆర్థర్. హీ కండక్టెడ్ సమ్ ఎక్స్పెరిమెంట్స్ ఆన్ రాబిట్. Okay, so he repeatedly gave the antigen injection. Okay, so to the rabbit. But when compared with the number of antigen, so more number of antibodies are produced. So that is called antibody excess. Okay, so then second time an injection, first time ఇంట్రావీనస్ ఇచ్చి సెకండ్ టైం సబ్క్యూటేనియస్ ఇచ్చినప్పుడు ఇంజెక్షన్ ఇచ్చినప్పుడు ఏమైంది? సివియర్ రియాక్షన్ అనేది జరిగింది. ఓకే, అల్టిమేట్లీ దట్ ర్యాబిట్ వాజ్ డైడ్. ఓకే, సో డ్యూ టు excess of production of antibodies, those excess of antibodies will go and accumulate or surround the the antigen, that leads to formation of a bigger immune complex in various tissues. Okay, so that leads to attraction of the phagocytes as it is. Okay, they engulf the self tissue also. Okay, so vascular accumulation and blood vessels are damaged. Okay, tissue damage are the major identification యాక్షన్స్. ఓకే, సో దీనివల్ల ఏమవుతుందంటే బ్లడ్ వెజల్స్ డ్యామేజ్ అవుతాయి. ఎందుకంటే ఈ యాంటీజెన్ యాంటీబాడీ కాంప్లెక్సెస్ దే ఆర్ మోస్ట్లీ సాల్యుబుల్ ఇన్ దిస్ ఎగ్జాంపుల్. దే కెనాట్ బి ఐదర్ ఐడెంటిఫైడ్ బై అవర్ ఓన్ ఇమ్యూన్ సిస్టమ్ బికాస్ దే ఆర్ సాల్యుబుల్ ఇన్ స్టేట్. సో దే కెనాట్ బి ఐడెంటిఫైడ్ ఈజీలీ బై అవర్ ఇమ్యూన్ సిస్టమ్ సెల్స్. సో దట్ లీడ్స్ టు డిఫరెంట్ టిష్యూ డ్యామేజ్ అవుతుంది. vascular accumulation. What is vascular accumulation? It is the meaning ఏంటి అంటే close. Okay, so accumulation అంటే closing of blood capillaries, vascular blood capillaries. Okay, accumulation means close. So vascular accumulation takes place. Right. Next, another example for type 3 hypersensitivity is serum sickness. So it is antigen excess. Antibody excess is the antigen excess. So due to that, ఇక్కడ ఏమవుతుందంటే నెంబర్ ఆఫ్ యాంటీబాడీస్ కన్నా యాంటీజెన్స్ నెంబర్ అనేది ఎక్కువగా ఉండడం నెంబర్ 1. ఓకే, సో నెంబర్ 2 ఏంటంటే దోస్ యాంటీజెన్స్. 1 సెకండ్. హలో అసినో. Let's continue. అయిపోయింది క్లాస్ దగ్గరికి వచ్చింది. రైట్, హియర్ antigen due to antigen excess than the antibodies and their size is bigger. Okay, and they are soluble in nature. They cannot be easily identified by our phagocytes. So these antigen antibody complexes, they are stored in kidneys mostly. Okay, that leads to fever, allergy, joint pains, lymph node swelling, and some kidney problems. Not severe, but some kidney problems are occurred. So it right. So దీనివల్ల కొన్ని డిసీజెస్ అనేటువంటి వస్తాయి. గ్లోమరులోనెఫ్రైటిస్, these are the some diseases, okay, that are occurred due to antigen excess of serum sickness. Okay, so glomerulonephritis, rheumatoid fever, dengue, hemorrhagic fever are some of the diseases that are occurred due to serum sickness. And then type 4, which is last one. So it was explained by scientists who it was explained by Coombs and Gell in 1963. సో ఇప్పటి వరకు అంటే లాస్ట్ 3 చెప్పుకున్నవి వాటి యొక్క యాక్షన్ అనేటువంటి ఈ వెయిటింగ్ పీరియడ్ తర్వాత సెకండ్ డోస్ ఇచ్చినప్పుడు ఏమవుతుందంటే సో విత్ ఇన్ హవర్స్ లో యాక్షన్ అనేది జరుగుతుంది. వెరీ ఫాస్ట్ గా జరిగేది టైప్ 1 ఆర్ అనఫైలాక్సిస్, విత్ ఇన్ మినిట్స్ లో జరిగిపోతుంది. ఓకే, బట్ టైప్ 4 hypersensitivity takes approximately 24 to 72 hours. So that's why it is also known as delayed type hypersensitivity. Hypersensitivity. Okay, so delayed type of hypersensitivity. రైట్. సో దీన్నే షార్ట్ ఫామ్ లో గుర్తుంచుకోండి, టీటీహెచ్ అనే అడుగుతారు ఎగ్జామ్ లో. ఓకే, డిలేడ్ టైప్ ఆఫ్ హైపర్ సెన్సిటివిటీ. రైట్. సో ఇట్ ఈస్ ఎక్స్క్లూజివ్లీ సెల్ మీడియేటెడ్. దేర్ ఆర్ నో రోల్ ఆఫ్ యాంటీబాడీస్ ఇన్ దిస్ కేస్. ఓన్లీ sensitized T helper cells, cytotoxic T helper cells, macrophages. Okay, all those are participated. So it is exclusively cell mediated type of hypersensitivity. Okay, in this T cells and macrophages are they mediate this hypersensitivity. It occurs generally in case of either tissue transplantation or some what we call the due to some chemicals also. Okay, so anyway, some T cells are generally sensitized to some sensitized to some antigens. Okay, so second time when same antigen entered, there will be a cooperation between the antigen and T cells occurred. Okay, so that leads to self tissue damage takes place. There are 2 examples for type 4 kind of hypersensitivity. One is tuberculin type of hypersensitivity and contact dermatitis. Okay, so sometimes during some experiments regarding some vaccines, okay, some live attenuated or sometimes purely live bacteria are injected to experimental animals or to human models also. Okay, in such conditions, okay, like mycobacterium tuberculosis, actinomyces, aspergillus, candida, penicillium. Okay, so in such conditions, what happens? They're already exposed or sensitized to these mycobacterium tuberculosis, actinomyces. If at all they are sensitized, those people are animal, aspergillus, candida, penicillium notatum, like fungi. So when we deliberately give more antigens or sensitized antigen is given, okay, so the tissue damage is generally occurred generally in lungs, wherever these pathogens they reside, so that tissue is damaged. Okay, so as it is first identified in tuberculosis experiments, so this reaction is called tuberculin type of type 4 hypersensitivity. And contact dermatitis. Okay, sometimes some chemicals, they cannot act as so it is also known as haptin mediated type of hypersensitivity. What is haptin? Haptin is generally not an antigen. It is not an immunogen. Okay, but whenever it is introduced by adding to some protein or larger molecule, so then it acts as an antigen. Okay, So here such chemicals are attached to our own antigens. ఇక్కడ కొన్ని కెమికల్స్ మనం ఇన్హేల్ చేసినప్పుడు, మనం ఇంజెస్ట్ చేసినప్పుడు ఏమవుతుందంటే, సో దీస్ కెమికల్స్ విల్ గో అండ్ బైండ్ టు అవర్ ఓన్ యాంటీజెన్స్. దెన్ దట్ కెమికల్ విల్ బికమ్ అన్ యాంటీజెన్, న్యూ యాంటీజెన్. సో మన యాంటీజెన్ కట్టుకోవడం వల్ల అవర్ ఇమ్యూన్ సిస్టమ్ ఏమనుకుంటుంది, సో దట్ దట్ రికగ్నైజెస్ that newly formed haptenic antigen as foreign, and here the sensitized T helper cells will reach there, will damage that haptenic cells, okay, which are self cells. Okay, so it causes cell mediated immunity. Okay, particularly in the skin it takes place. Can anybody tell what are Langerhans cells?
Antigen presenting cells.
Very good. Who is that?
Kavita, sir.
Very good. Okay. So Langerhans cells are a kind of antigen presenting cells. They are dendritic cells, which are present in the skin. Okay. So they actively participate here. Okay. They damage our own skin cells. Okay. So that leads to some allergy type of rashes occurred on our skin. Okay, so due to chemicals like nickel, chromium salts, formaldehyde, hypo like photography developing chemical, okay, they act as haptens on our skin whenever we touch it. Okay, so this reaction takes place, but it takes time up to 24 to 72 hours. So as it is delayed time. Okay, so these are the kind of type of hypersensitivity reactions: type 1, type 2, type 3, type 4. Here contact dermatitis takes place 40%. It causes eczema. Tuberculin type of reaction causes local induces local cell damage, tissue damage, and granuloma. Okay, so a kind of hardening of the tissue. Okay, so all these occur this much time. That's why this is a delayed type of hypersensitivity. What you should remember is type 1 anaphylactic reaction. Okay, so also known as atopic fever or immediate type of hypersensitivity. That is type 1. And the type 2 antibody dependent cell mediated cytotoxicity, or what we call the isoimmune reactions and autoimmune diseases and drug-induced hypersensitivity. Okay, or erythroblastosis fetalis. These examples you should remember for the type 2. And then type 3 is immunocomplexes mediated hypersensitivity. And type 4, type 3 lo malli Arthur's reaction and serum sickness, antibody excess, antigen excess. That's good to score. Just type 4 delayed type of hypersensitivity, DTH. Okay, so ఇది గుర్తుంచుకోవాలి. It is cell mediated exclusively. అది కూడా గుర్తుంచుకోవాలి. And here tuberculin type of type 4 contact dermatitis type. ఇవి రెండు కూడా headings గుర్తుంచుకోవాలి. Right? So with this, we almost completed, the immunology. But ఇంకొక టాపిక్ అది సిలబస్ లో లేదు, మనకు జేఎల్ సిలబస్ సైటోకైన్స్ టాపిక్. బట్ అది దట్ ఈస్ పార్ట్ ఆఫ్ ఇమ్యూనాలజీ. రేపు ఒక 20 minutes కంప్లీట్ చేసి ఇమ్మీడియట్ గా రేపు నేను ఐ స్టార్ట్ ఫిజియాలజీ. ఓకే, సో రేపటి నుంచి అవర్ ఓన్ జువాలజీ కాబట్టి ఇంకా రెగ్యులర్ గా అటెండ్ కండి. అండ్ నేను ఈ వీక్ లో ఏదో రోజు సెలవు పెడతాను, అంటే వర్క్ లో బయటకు వెళ్తాను. అప్పుడు నేను ఈ రోజు టాపిక్ అండ్ నిన్న టాపిక్ పైన టెస్ట్ కండక్ట్ చేస్తాను. 2 టెస్ట్ సెపరేట్ టెస్ట్ కండక్ట్ చేస్తాను. ఏ రోజు ఒకవేళ నేను రాకపోతే సండే క్లాస్ తీసుకుంటాను. వర్కింగ్ డే రోజు కనుక నేను అబ్సెంట్ అయితే దట్ విల్ బి కవర్డ్ ఆన్ సండే. 1 అవర్ కానీ 2 అవర్ కానీ. ఒకవేళ నేను 2 days రాలేదంటే సండే రోజు 2 క్లాసెస్ తీసుకుంటాను, మార్నింగ్ ఒకటి ఈవినింగ్ ఒకటి. రైట్. సో అండ్ ఓకే, ఐఎమ్ క్లోజింగ్. మోస్ట్ ఆఫ్ యూ హ్యావ్ పేడ్ ఫీ. ఈ రోజు జాయిన్ అయిన వాళ్ళు చాలా మంది దాదాపు పే చేశారు. ఎవరైనా ఇఫ్ ఎట్ ఆల్ ఇంకా మర్చిపోయి ఉంటే